Welcome back, everybody. It's time for another deep dive. And today we're going to be taking a close look at the proximal tubule.
Oh, excellent.
We've got this chapter from a nephrology textbook to guide us. I know you're a medical pro, so we can skip those basic definitions and analogies, right?
Yeah, let's cut to the chase.
Perfect. So the proximal tubule,
Yeah,
it's a workhorse.
it is.
The text says it reabsorbs something like 65% of the glomerular filtrate,
Oh.
and it's all isosmotic to plasma.
Yeah, that's a huge amount to process. It's really key to remember that the composition of what's being reabsorbed is essentially the same as plasma. That's how osmotic balance is maintained.
Right. But it's not just indiscriminate reabsorption.
Right. No, not at all.
The text mentions some specific percentages.
Uh-huh.
Bicarbonate reabsorption is around 90% sodium, 65%,
Right.
and chloride, 55%.
Exactly. And we can't forget about secretion. It's happening at the same time.
Oh, right. There's stuff going in and out constantly.
Absolutely. Things like hydrogen ions and a bunch of organic anions and encharations. Those are all being secreted into the tubule.
So it's a two-way street, not just reabsorption.
Definitely a dynamic process. Now let's talk about the structure of the proximal tubule itself.
Yeah. The source mentions convoluted and straight segments, and then further divides it into S1, S2, and S3 segments.
You got it. And each of these segments has its own unique functional characteristics.
Okay. So we're not dealing with just a uniform tube here.
Nope, not at all. And get this, monoclonal antibodies can actually be used to isolate these segments.
Really?
Yeah, because they express these unique cell surface peptidases. It's incredibly precise.
Wow. So how do these segments differ functionally? I mean, what are the key differences?
Well, the S1 segment, for example,
Oh,
it's got a really high capacity for sodium and bicarbonate reabsorption.
makes sense, I guess. That's where most of the filtrate enters.
Exactly. So reclaiming those essential substances early on is crucial.
Right. And the S2 segment,
That's where you find the most prominent organic anion and kination secretory pumps.
like a specialized waste disposal system.
Precisely. Now let's get to the heart of it all, the transport mechanisms.
I was just thinking about that. The text emphasizes the central role of active sodium transport.
Right. It's the primary event in proximal tubular function.
So why is sodium so crucial?
Well, because the active transport of sodium, it sets off this whole cascade of events, and that ultimately leads to the passive reabsorption of water and a bunch of other filtered solutes. Think of it like a domino effect.
Okay. So sodium is the first domino that sets everything else in motion.
Exactly. And the text also talks about the sodium-potassium AT pace pump.
Yeah, that's essential for maintaining that low intracellular sodium concentration.
You got it. And that low concentration is what allows sodium to keep flowing in, driving everything else.
Okay, so sodium's the key. And that low intracellular concentration is maintained by the sodium-potassium ATPase pump.
Right. And that forms the basis for secondary active transport.
Secondary active transport.
Yeah, where sodium's movement down its concentration gradient, it provides the energy to move other substances against their concentration gradients.
Like what kind of substances?
Well, things like glucose amino acids and phosphate.
So sodium's pulling these important substances along for the ride.
Precisely. Now, another interesting point is how the reabsorption of sodium bicarbonate early in the proximal tubule, it actually ends up increasing the chloride concentration in the tubular fluid.
Interesting. So then that higher chloride concentration drives passive sodium chloride reabsorption later on.
Exactly. It's like a chain reaction all stemming from sodium.
Right. And it's not just simple diffusion either.
No. There are neurohumoral factors that can modulate these processes too.
Like angiotensin II and norepinephrine.
Yes, they stimulate sodium chloride and water reabsorption.
And dopamine actually does the opposite. It inhibits reabsorption.
That's right. It highlights just how complex these regulatory mechanisms really are. And then, of course, we have the peritubular capillary hemodynamics.
Yeah. Those influence fluid reabsorption too.
They do. And then there's this concept of glomerulotubular balance.
Right, where even when the GFR changes, fractional tubular reabsorption stays pretty much constant.
It's amazing how the proximal tubule adapts to maintain that balance.
Yeah. It really speaks to the kidney's remarkable ability to self-regulate. So we've talked about sodium and its central role.
Yes.
But what about the handling of other specific solutes?
Right. We've got to look at how sodium reabsorption is intertwined with the reabsorption or secretion of other solutes, but each one has its own unique story.
Okay, so sodium is our main character. But there's a whole supporting cast of solutes.
Exactly.
Where should we start?
Well, let's start with bicarbonate. We talked about how the proximal tubule reabsorbs a large portion of filtered bicarbonate, and the source goes into the mechanism behind that. And it also highlights how volume depletion and expansion can impact bicarbonate reabsorption.
Okay, so what about glucose?
So glucose reabsorption, it's
Right.
primarily mediated by the sodium-glucose cotransporter.
Right, which relies on that sodium gradient,
Exactly, but there's a limit to how much glucose this transporter can
the
handle.
transport maximum.
That's it, the TB.
What happens when that limit is exceeded?
Well, when the filtered load of glucose goes above the 2, you start to see glucose spill into the urine.
Glucosuria.
You got it. And this is commonly seen in uncontrolled diabetes mellitus.
Right, where the high blood sugar overwhelms the transporters.
Exactly. So glucose reabsorption, it's directly tied to sodium and has some important clinical implications.
Okay. What about urea?
Urea reabsorption is largely passive. It's driven by water reabsorption.
So as water is reabsorbed, the urea concentration increases.
And that creates a gradient that favors its passive reabsorption,
Following water.
pretty much.
Yeah.
Now, for calcium,
Okay.
most calcium reabsorption in the proximal tubule is also passive, moving along with sodium chloride and water.
Right. And then parathyroid hormone and vitamin D play a role in regulating calcium reabsorption in later parts of the nephron.
Exactly. The interplay between sodium chloride transport and calcium reabsorption is particularly important clinically.
Interesting. So sodium handling can indirectly affect calcium levels.
It can,
Mm-hmm.
and that can have consequences.
Anything else about calcium reabsorption?
Well, the text also mentions the role of phosphate.
Okay.
Sodium phosphate cotransporters are involved in phosphate reabsorption,
Right.
and PTH regulates that process too. It's another example of how interconnected everything is in the proximal tubule.
Right. We've got sodium glucose, urea, calcium, and phosphate all interacting in this one segment of the nephron.
That's right. It's quite a busy place. Now, what about magnesium?
Yeah.
So the source focuses on the passive reabsorption of magnesium in the thick ascending limb,
Okay.
and it's facilitated by this tight junction protein called paracellin 1.
Paracellin 1.
Yeah. And this process is influenced by the electrochemical gradient in the thick ascending limb, which can be affected by things like loop diuretics.
Interesting. So those medications can actually increase magnesium excretion.
Yeah, sometimes causing hypomagnesemia. So we see that both acquired and genetic factors can disrupt magnesium handling.
Wow, it's amazing how manipulating sodium handling can have ripple effects on so many other electrolytes.
It really highlights the complexity of renal physiology. And we haven't even touched on urate organic anions and cations or protein handling yet.
You're right. There's so much more to uncover.
Yeah.
We'll have to delve into those other substances in part 2 of this deep dive. All right, so we've seen how the proximal tubule handles all these different salts, but I'm really curious about the clinical implications. How does understanding all of this actually help us in practice?
Well, grasping these intricate mechanisms, it's really crucial for interpreting clinical presentations and also those puzzling lab values we often encounter.
Right, abnormal lab results. They often point to a dysfunction somewhere in the proximal tubule.
Exactly. They can be like a clue.
Can you give me some examples?
Oh, absolutely.
Hmm.
Let's go back to glucose for a minute.
Okay.
We talked about how exceeding that transport maximum leads to glucosuria in diabetes.
Right.
But recognizing glucosuria can also help diagnose other conditions like renal glucosuria.
Yeah, text mentioned that. What's the significance of that?
Well, in renal glucosuria, glucose spills into the urine even when blood sugar levels are normal.
Oh, so it's not diabetes.
Right. It suggests a problem with the proximal tubule's ability to reabsorb glucose, maybe due to a defect in those sodium-glucose cotransporters.
So it's like a clue pointing us toward a specific dysfunction.
Exactly. And then there's Fanconi syndrome.
Fanconi syndrome.
It's a generalized disorder of proximal tubular transport.
Oh, wow.
Yeah. It can cause a whole bunch of abnormalities all at once. Glucosuria, aminoaciduria, phosphaturia. It's like the proximal tubule just can't hold on to anything.
That sounds pretty serious.
It can be. It really underscores how interconnected all of these transport mechanisms are.
So a single syndrome can disrupt multiple solute handling systems.
Yeah. Now let's switch gears to urea for a second.
Okay.
The text points out that BUN levels often vary inversely with GFR.
Right. If less urea is filtered, BUN levels rise.
Exactly. But there are exceptions to that rule.
Like when urea production increases.
Like in gastrointestinal bleeding.
Right. Or in volume depletion.
Yeah, in volume depletion, you get increased proximal sodium and water reabsorption, which actually enhances urea reabsorption too.
So that also causes a rise in BUN.
Exactly. The text calls this prerenal azotemia,
A prerenal azotemia.
meaning that the elevated BUN is not due to intrinsic kidney disease. It's due to factors affecting blood flow to the kidneys.
An important section.
Very important. Okay. Back to calcium and its relationship with sodium handling.
Yeah, we were talking about that before.
Right. So remember how most calcium reabsorption in the proximal tubule and loop of Henle, It's passive. It's driven by sodium chloride and water reabsorption.
Mm-hmm.
Well, that has some important clinical implications.
Meaning?
Well, anything that affects sodium reabsorption in those segments, it's going to affect calcium reabsorption too.
So conditions like volume depletion or
Exactly.
diuretics acting in the proximal tubular loop of Henle.
Right. Those can all potentially lead to increased calcium excretion.
And that could increase the risk of calcium stones.
Exactly, which are discussed later in the text.
Does the text say anything about how we can address that clinically?
It does. It explains how medications can actually manipulate this sodium-calcium link for therapeutic benefit.
Interesting.
For example, thiazide diuretics. They act in the distal tubule,
Okay.
and they can actually lower calcium excretion
Boy.
by increasing sodium and subsequently calcium reabsorption in earlier segments of the nephron.
So we're using our understanding of these mechanisms to fine-tune solute reabsorption.
Precisely. Now, the source mentions another clinically relevant scenario. This one involves organic cations.
Oh yeah, the competition between organications for those secretory pathways.
Right. The text explains how medications like cimetidine, they can actually compete with creatinine for secretion.
And since we use creatinine to estimate GFR
Yeah, this competition can falsely elevate serum creatinine levels.
without actually reflecting a decreased GFR.
Exactly. That's something to watch out for.
Definitely. Now, what about phosphate?
Okay, so with phosphate, the text highlights the role of PTH in regulating phosphate reabsorption.
Right.
So increased PTH, it leads to decreased phosphate reabsorption and increased excretion.
Which makes sense because when calcium levels are low, PTH is released to increase calcium reabsorption.
Exactly.
But that often comes at the expense of phosphate excretion.
Precisely. And the text also mentions how metabolic acidosis can affect phosphate reabsorption.
It said that transporter activity is diminished in acidosis.
Right. Leading to increased phosphate excretion.
And that phosphaturia is actually beneficial in acidosis.
It is. It helps excrete more acid buffers.
So the kidneys are adjusting to maintain that acid-base balance.
Exactly. Now, for magnesium,
Yes.
most magnesium reabsorption happens in the thick ascending limb,
Right.
primarily through the paracellular pathway.
Mm.
And remember that tight junction protein paracellin 1?
Yeah.
It plays a key role in facilitating that passive reabsorption.
Right. And anything that influences the electrochemical gradient in the thick ascending limb can affect magnesium reabsorption.
Exactly, like those loop diuretics we talked about.
Right? By inhibiting sodium and chloride reabsorption, they can increase magnesium excretion.
Potentially leading to hypomagnesemia.
And the text also mentioned genetic mutations in the paracellin 1 gene.
Right. Those can cause a rare inherited disorder characterized by magnesium wasting.
So both acquired and genetic factors can disrupt magnesium handling.
That's right. Now, what about urate?
Yeah, we were talking about that complex handling process earlier.
Right. Remember that three-step process in the proximal tubule?
Reabsorption, secretion, and post-secretory reabsorption.
You got it. It's a delicate dance that ultimately determines how much urate is excreted.
And those processes are influenced by proximal sodium transport and volume status.
Exactly. So in volume depletion, for example.
You often see a decrease in both sodium and urate excretion.
That's right. Sodium just keeps popping up everywhere.
It really does. It seems to be connected to everything.
It really is. This link between sodium handling and urate excretion, it's especially important in conditions like gout.
Right, where elevated urate levels lead to all that painful joint inflammation.
That's exactly. Okay, let's move on to organic anions and their secretory pathways.
Okay.
So the text highlights the role of anion exchangers, explaining how organic anions are taken up into the cell at the basolateral membrane and then secreted into the tubular lumen.
Right. And it mentioned how albumin binding influences the secretion of certain organic anions.
Yes, albumin binding actually facilitates secretion by delivering those substances to the secretory sites in the kidney.
Yeah.
It's like a targeted delivery system, maximizing their excretion.
So it helps get rid of them.
Exactly. And this process is crucial for eliminating certain medications and toxins that are highly protein-bound.
The text used the example of salicylate intoxication.
Right, where alkalinizing the urine enhances salicylate excretion.
By reducing its reabsorption.
Exactly. So we're highlighting the clinical importance of understanding these secretory pathways and how manipulating urine pH can influence drug elimination.
So we're harnessing the kidney's own mechanisms to enhance drug clearance.
Precisely. Now, before we move on, what about protein handling in the proximal tubule?
Yeah, how does that work?
Well, the text explains how small proteins and peptides, they're filtered and then reabsorbed via endocytosis.
Okay.
And this reabsorption serves two main purposes:
What are those?
preserving nitrogen balance and participating in hormonal homeostasis.
So the kidneys aren't just filtering waste products.
No, they're reclaiming valuable substances
Yeah,
and helping regulate hormone levels.
that makes sense. Is there anything else about protein handling that we should know?
The text mentions how albumin, when it's filtered in greater than normal amounts, it can be reabsorbed by a separate low-affinity, high-capacity endocytic process.
And when does that happen?
Well, this is commonly seen in nephrotic syndrome.
Nephritic syndrome.
Yeah, where increased reabsorption and subsequent catabolism of filtered albumin, they contribute to the hypoalbuminemia often seen in this condition.
So the proximal tubule can adapt,
Hmm.
but sometimes with unintended consequences.
Exactly. So we've covered a lot of ground here, highlighting how the physiological mechanisms of the proximal tubule, they directly impact clinical practice.
We've learned about interpreting those abnormal lab values, recognizing drug interactions, and optimizing treatment strategies.
Exactly. Appreciating these complexities is really paramount.
Absolutely. Are there any other Solutes or clinical scenarios we should discuss before moving on? You mentioned a lot of different Solutes and their clinical implications, but there's one that the text covers that I found particularly interesting.
Oh, yeah. Which one?
Citrate.
Ah, citrate.
It's not the first thing that comes to mind when I think about renal function,
Right?
but it seems like it plays a pretty crucial role.
It does. It's actually fascinating. So the text mentions that most filtered citrate is reabsorbed in the proximal tubule, and this reabsorption is linked to sodium.
So sodium again.
Of course, sodium's always involved. Citrate enters the proximal tubular cells via a sodium citrate cotransporter.
So again, highlighting the importance of that sodium gradient.
Exactly. Sodium is the key. What makes citrate's reabsorption so unique is that it's influenced by acid-base balance.
Oh, right. In metabolic acidosis, citrate reabsorption decreases, leading to increased excretion.
Citrate acts as a buffer. So excreting more helps get rid of that excess acid.
The kidneys are so good at adapting.
They really are, always adjusting to maintain that balance. Clinically, citrate handling is particularly relevant when it comes to calcium stone formation.
Right. The text explains how citrate can inhibit the precipitation of calcium oxalate and calcium phosphate, the main components of kidney stones.
Exactly. It binds to free calcium, forming a soluble complex that's less likely to crystallize.
So adequate citrate levels in the urine can actually help prevent stone formation.
That's the idea. And
Yeah.
that's why hypocitraturia, or low urinary citrate, is a risk factor for stone disease.
Makes sense. And the text even mentions citrate supplementation as a therapeutic option.
Yeah, to increase urinary citrate and hopefully prevent stones.
Another example of how understanding the physiology leads to practical treatments.
Absolutely. Now, we've talked about a lot of different transport mechanisms, but what about those secretory pumps?
You mean those organic anion and concation secretory pumps?
Yeah, those are the ones. They're responsible for secreting a wide variety of substances.
Right. And they use energy from ATP to move those organic anions and cations from the blood into the tubular lumen.
Exactly. It's an active process. Unlike the passive reabsorption we talked about for a lot of the other selenium.
And they're essential for getting rid of waste products, toxins, and even medications.
You got it. The text also mentioned competition for these pathways
Oh, yeah.
between different substances.
Exactly. Which can lead to potential drug interactions. It's like they're all vying for a spot on the transport system.
Great analogy. Clinically understanding these interactions is so important for predicting how different medications might affect each other's clearance.
Couldn't agree more. So to wrap up, we've explored the intricate workings of the proximal tubule, from its impressive reabsorptive capacity to those very specific secretory pathways.
And we've seen how sodium plays a central role in so many of these processes.
Absolutely. Sodium influences the handling of a really diverse array of solutes.
And we've also seen how all of these mechanisms directly impact clinical practice. They do, helping us interpret lab values, recognize drug interactions, and develop effective treatments.
It's really amazing how the kidney can maintain such a delicate balance.
It's a testament to its complexity and efficiency.
As we wrap up this deep dive into the proximal tubule, remember how the kidney's intricate mechanisms for solute handling underscore just how delicate that balance needs to be for overall health? So keep exploring, keep asking questions, and keep diving deep into the fascinating world of nephrology. Thanks for joining us.